All right, so moving on to our 4.3, our non-proteobacteria, gram-negative bacteria, and then also we'll talk about the phototrophic bacteria. So the majority of the gram-negative bacteria belong to the phylum Proteobacteria. So remember we talked about that phylum Proteobacteria just a moment ago. We talked about all of the 5 different groups in that Proteobacteria. So the majority of the gram-negative bacteria are a part of that phylum. And we talked about those in detail. Now we're going to talk about those that are non-proteobacteria. So in this section we're going to describe the 3 classes of gram-negative non-proteobacteria. So the Spirochetes, that's one, the CFB group, and then we are going to talk about Planktomycetes, which are actually a diverse group of phototrophic bacteria that do include some proteobacteria and also non-proteobacteria. So we'll talk about that as well. So first we're going to talk about our Spirochetes here. So Spirochetes are characterized by their long spiral-shaped bodies, and we've already looked at some Spirochetes in previous chapters when we were identifying cell morphology. Most Spirochetes are also very thin, which makes it difficult to examine gram-stain preparations under a conventional bright field microscope like we would see in the laboratory. Dark field fluorescent microscopy is typically used instead. So so we can just shine a light. Remember, the dark field has the dark background, and then we can shine a light at an angle toward these little Spirochetes, and then they will light up, the fluorescent microscopy and the dark field. So Spirochetes are also difficult or even impossible to culture. So these guys are really difficult. They are highly motile, so they move around a lot, and they use what's called an axial filament to propel themselves. So they don't have a flagellum like we've discussed with other bacteria, but these have what's called an axial filament, and we'll look at that in just a moment. The axial filament is similar to a flagellum, but it wraps around the cell and runs inside the cell body of a spirochete. So in that periplasmic space between the outer membrane and the plasma membrane. So let's take a look at that. We'll see the picture here. So here's our dark field microscope image. So you can see the background is very dark in color, and then we have our very light spirochetes there, and you can see how spiral they look, very wiggly. Here we have a TEM image, and then you can see it's actually a cross section. So it's like if you took one of those long spirochetes and just cut it right in the middle and looked down the spirochete. And you can see here the axial filament is actually in this area here. You can see that this is the cell membrane, and then we have the periplasmic space, which is generally very small in this area here, but is very lumped out in this area because we have the axial filament there. And then there's the illustration to make it a little bit more clear. So we have our inner membrane, the cell membrane there, and then we have the outer membrane, and then we have the periplasmic space in between, and then on the one side here we have the axial filament. And then you can also see in the drawing over here, the nucleoid region is going to be in this area here, and then the cytoplasm, the lighter colored area here. And then you can see on this SEM here, the other electron micrograph, that you can actually notice this, this section here is part of it wrapped around, and you can see obviously better in the illustration here. So we've got this kind of wrapping around that goes along the whole length of the spirochete in that periplasmic space, and then it's going to twist in order to allow these guys to kind of squiggle along and keep moving. So definitely different than a flagellum, but also gives movement to that spirochete. So there are several genera of spirochetes that include human pathogens. For example, Treponema includes the species Treponema pallidum, which is further classified into 4 subspecies. So the Treponema pallidum, Treponema pallidum perteniu, Treponema pallidum caratium, Treponema pallidum endemicum, or endemicum. The subspecies, pallidum pallidum, so Treponema pallidum, causes the sexually transmitted infection known as syphilis. So when we're talking about Treponema groups, oftentimes you can think of Treponema pallidum, which gives the infection of syphilis. So the third most prevalent sexually transmitted bacterial infection in the United States, after chlamydia, which we talked about, and gonorrhea, which we talked about. So the other species of Treponema pallidum cause tropical infection diseases of the skin, bones, and joints. All right, so the pallidum, you can remember, is going to cause syphilis, and then all of the other ones are going to cause tropical infection diseases of the skin, bones, and joints. Another genus of spirochetes is Borrelia. It contains a number of pathogenic species. For one, we have Borrelia burgdorferi, which causes Lyme disease, which is transmitted by several genera of ticks. So the ticks actually carry the Borrelia burgdorferi, and then when the tick bites someone or gets its head stuck in someone, then it transmits that. It's going to pass along that bacteria, the Borrelia burgdorferi, the spirochete, and then that will cause Lyme disease in the human or the animal. So it often produces a bullseye rash, like a red, it looks like a bullseye with the lines around it type of rash, a fever, fatigue, and then sometimes debilitating arthritis. If you notice that you have a tick and you are concerned that you might have Lyme disease, you can notice and pay attention and see if this bullseye rash comes up in the first, I think it is like 36 hours. If it does, you can go to an urgent care or a doctor, and then you can take some medicine to help try to make sure that you don't have Lyme disease, because then otherwise it can stay with you for life sometimes. Then there's also Borrelia recurrens, which causes a condition known as relapsing fever. So recurring, relapsing fever. And then again, we have our Appendix D is going to list some genera, species, and related diseases for spirochetes. So we're going to move on to the next group. So again, if we back up a little bit, let's remember what we're working on here. Remember I mentioned these learning objectives are helpful for that. So we're going to be talking about non-proteobacteria, which again are gram-negative bacteria. And then we're going to be looking at the different categories. Okay, so when we're looking about non-proteobacteria, we're going to be talking about spirochetes, the CFB group, and the planktomycetes. So we just talked about the spirochetes. Now we're going to move on to our CFB group, which stands for Cytophaga Fusobacterium Bacteroides, so CFB. Again, gram-negative, non-proteobacteria. Cytophaga Fusobacterium Bacteroides are classified together as a phylum and called the CFB group. So that is the phylum, is the CFB group. Although they are phylogenetically diverse, bacteria of this CFB group do share some similarities in the sequence of their nucleotides and their DNA. So remember, when we are going through, most recently in modern times, we're going through and looking at the DNA nucleotides of bacteria in order to classify them. So in this classification, these have some similarities. The C, the F, and the B group have similarities that made it so that scientists grouped them together in this way. They are rod-shaped bacteria. They are adapted to anaerobic environments, remember, without oxygen environments, such as the tissue of the gums, the gut, and rumen of ruminating animals. The CFB bacteria are avid fermenters. They're able to process cellulose in rumen, thus enabling ruminant animals to obtain carbon and energy from grazing. So remember, cellulose is something that oftentimes animals are not able to break down. So we have bacteria in our intestines that can do that for us. Cytophaga are motile aquatic bacteria that glide. Fusobacteria inhabit the human mouth and may cause serious infectious diseases. So the largest genus of the CFB group is Bacteroides. So we're going to talk more about that. So very briefly here, Cytophaga, what you need to know about that, they're motile aquatic bacteria and they glide. So they're able to glide in movement. Fusobacteria are in the human mouth and can cause some infectious diseases. And then we'll look at the chart down here in the table in a moment. The largest of the group then is Bacteroides, which includes dozens of species that are prevalent inhabitants of the human large intestine, so making up about 30% of the entire gut microbiome. So again, when we're talking about Our microbiome, that's all of the cells, all of the microorganisms that we find in a human. In specifically, we're talking about the human gut here. And about 30% of the bacteria, of the microorganisms in the human gut, are Bacteroides. So 1 gram of human feces contains up to 100 billion Bacteroides cells. Most Bacteroides are mutualistic. So remember, mutualistic is beneficial for both. They get something out of it, we get something out of it. They benefit from nutrients they find in the gut, things that we've eaten, and the human benefits from their ability to prevent pathogens from colonizing the large intestine. So when we have lots of Bacteroides, high populations of Bacteroides in the gut, then that is going to prevent other bacteria from coming in and inhabiting the gut, because all of the nutrients are already being used and there's already so many of this bacteria that other things can't kind of set up shop because there's not the space to do that. However, when the populations of Bacteroides are reduced, which happens if someone takes antibiotics, then the gut becomes a more favorable environment for other pathogenic bacteria, including fungi, which can cause secondary infections. So again, taking antibiotics, especially those broad spectrum antibiotics, will go through and kill everything it can reach, including our gut, which has about 30% of which are Bacteroides. And when those are wiped out, then we're vulnerable. We have this space, this ability for other things to move in, other pathogenic bacteria and fungi to move in. Only a few species of Bacteroides are pathogenic. B. melanogenicus, I'm not familiar with that one so much, for example, can cause wound infections in patients with weakened immune systems. All right, so then we're going to move into this last group here, the planktomycetes. So the planktomycetes are found in aquatic environments, inhabiting fresh water, salt water, and brackish water. So fresh water, meaning without salt, the kind of water we could drink. Then salt water, like the ocean, highly salty. And then brackish is a mix between the two. Planktomycetes are unusual in that they reproduce by budding, meaning that instead of one maternal cell splitting into 2 equal daughter cells, which is called binary fission, which is what we will talk about regarding bacteria. Most bacteria go through binary fission, which is where they just divide the cells evenly, one mother cell dividing into 2. The mother cell forms a bud that then detaches from the mother cell and lives as an independent cell. So it has a budding off process we see in these planktomycetes. These so-called swarmer cells then are motile and not attached to a surface. So we have this mother cell that's attached to a surface, and then it's going to bud off an independent cell, which is called a swarmer cell, and then it is motile. It can move around. It's not attached to a surface. However, they will soon differentiate into sessile or immobile cells with an appendage called a holdfast, and then that is going to allow them to attach to surfaces in the water. Only the sessile cells are able to reproduce. So that's kind of the life cycle there. We have our cell here, which you can see in A, is our sessile planktomycete. So we have one individual cell here, and then here's its holdfast. So it has this appendage that allows it to attach to something in the water. Allows to adhere to surfaces in aquatic environments. And then B, here's a swarmer, which is motile and lacks a holdfast. But then, so it'll float around, get to a spot, and then it'll grow this appendage, and then it'll have a holdfast and attach to something else. And then once it does that, it is a sessile planktomycete, which means then it can go through the budding process and make another one of these mobile cells, a swarmer cell. So here's our table for some non-Proteobacteria. Bacteroides you should be familiar with. Our Cytophaga, Fusobacterium, Leptospira, and some other ones are Treponema, etc. All right, so then we're going to look at the Phototrophic Bacteria. Okay, so remember going back up here, once again, 4.3, So we're talking about non-Proteobacteria, which again are gram-negative, and we're looking at the three groups of our non-Proteobacteria. So the three classes of gram-negative non-Proteobacteria. So the Spirochetes, the CFB group, and the Planktomycetes. And those are the three that we just talked about. And then we're going to look at our Phototrophic bacteria, which includes both Proteobacteria and non-Proteobacteria. Okay, so we've got those three groups that we talked about, non-Proteobacteria. Now we're going to look at something that's a combination that has some Proteobacteria and some non-Proteobacteria. And these are lumped in together as Phototrophic bacteria. So phototrophic bacteria are a large and diverse category of bacteria. They do not represent a taxon, but rather a group of bacteria that use sunlight as their primary source of energy. So phototrophic, think of photosynthesis, they use the sun as their primary source of energy. This group contains both Proteobacteria and non-Proteobacteria. They use solar energy to synthesize ATP through photosynthesis. When they produce oxygen, they perform oxygenic photosynthesis. When they do not produce oxygen, they perform anoxygenic photosynthesis. An meaning without oxygenic, without oxygen photosynthesis. So when they do produce oxygen, it's called oxygenic photosynthesis. When they do not, it's called anoxygenic photosynthesis. With the exception of some cyanobacteria, the majority of phototrophic bacteria perform anoxygenic photosynthesis. So when we talk about phototrophic bacteria, you can think mostly it's anoxygenic, so without producing oxygen. However, some cyanobacteria produce lots of oxygen, so they would be the oxygenic photosynthetic organisms. So one large group of phototrophic bacteria includes the purple or green bacteria that perform photosynthesis with the help of bacterial chlorophylls, which are green, purple, or blue pigments similar to chlorophyll in plants. So again, we're talking about photosynthesis. If you recall when you studied plants and plant cells, photosynthesis with chlorophylls, chlorophyll is an important part in the process of going through photosynthesis for a plant cell. We see something similar here. We have bacterial chlorophylls, which is similar to the chlorophyll in plants, and we can see that they're green, purple, or blue pigments. Some of these bacteria have varying amounts of red or orange pigments, then also called carotenoids. So we have chlorophylls, green, purple, blue pigmented chlorophylls, or bacterial chlorophylls. Some of the bacteria then have red or orange pigments called carotenoids. Their color varies from orange to red to purple to green. And they're able to absorb light of various wavelengths. Traditionally, these bacteria are classified into sulfur and non-sulfur bacteria. So let's take a look. And then they're further differentiated by their color. So even in this illustration here, this photo in this case, we have purple and green sulfur bacteria here that are using the bacterial chlorophylls to perform photosynthesis. So you can see the darker area here with our purple bacteria, and then the brighter green area with our green bacteria. The sulfur bacteria perform anoxygenic photosynthesis, right? So without oxygen, it doesn't produce oxygen, using sulfites as electron donors and releasing free elemental sulfur. All right, so the electron donors, they're donating electrons. The sulfites donate electrons to go through the process of photosynthesis. Remember, moving electrons to create energy. And then what that results in is free elemental sulfur, because we are taking the electrons from the sulfites, and then we have our sulfur. Non-sulfur bacteria use organic substrates such as succinate and malate as donors of electrons. So again, when we're talking about sulfur bacteria, they use sulfites and produce sulfur. The non-sulfur bacteria use organic substrates like succinate and malate for their electron donors. All right, so let's take a look at our different colors of our bacteria, of our sulfur bacteria and then our non-sulfur bacteria. So first we have purple sulfur bacteria. So they oxidize hydrogen sulfide into elemental sulfur and sulfuric acid. And then they get their purple color from the pigments, bacterial chlorophylls, and carotenoids. Okay, so again, since it's a sulfur bacteria, we're going to be taking a sulfite, or a sulfide in this case, hydrogen sulfide, and turning it into elemental sulfur, as well as sulfuric acid. They have their purple color from the bacterial chlorophylls and the carotenoids. Bacteria of the genus Chromatium are purple sulfur gamma proteobacteria. Okay, so again, remember we're talking about our phototrophic bacteria, which include both proteobacteria and non-proteobacteria. In this case, we have our gamma proteobacteria, Chromatium, which is a purple sulfur bacteria. These microorganisms are strict anaerobes, so without oxygen, and live in water. They use carbon dioxide as their only source of carbon, but their survival and growth are possible only in the presence of sulfites. So again, because we're talking about purple sulfur bacteria, which they use as electron donors. So Chromatium has been used as a model for studies of bacterial photosynthesis since the 50s. So highly used, highly looked at. So our purple sulfur bacteria, they go through photosynthesis, Chromatium. Then we can see our green sulfur bacteria. They use sulfide for oxidation and produce large amounts of green bacterial chlorophyll. Okay, so we don't have as much of the carotenoids, which remember are the red and orangish colors. So in this case we have bacterial chlorophyll. The genus Chlorobium is a green sulfur bacterium that is implicated in climate change because it produces methane. So remember at the very, very beginning of the chapter we talked about some environmental concerns with some bacteria, and this is one of those that is being looked at because it produces methane, which is a greenhouse gas. These bacteria use at least 4 types of chlorophyll for photosynthesis. The most prevalent, the bacterial chlorophyll, is stored in a special vesicle-like organelle called chlorosomes, and we talked about those in previous chapters as well. So when we talked about photosynthetic bacteria, we talked about chlorosomes, which were kind of like vesicles that contain these bacterial chlorophylls. Purple non-sulfur bacteria, which is our next group. So we looked at purple sulfur bacteria up here at the top. Now we're going to look at purple non-sulfur bacteria. Are similar to purple sulfur bacteria except that they use hydrogen rather than hydrogen sulfide for oxidation because it's non-sulfur, so it doesn't use the sulfide. Instead, it just uses the hydrogen. So among the purple non-sulfur bacteria is the genus Rhodospirillum. These microorganisms are facultative anaerobes, which are actually pink rather than purple, and they can metabolize or fix nitrogen. So these are some nitrogen fixers. They may be valuable in the field of biotechnology because they could possibly produce biological plastic and hydrogen fuel. So these are really looked at as a type of bacteria to see if we can produce some biological plastic. All right, then lastly, we have our green non-sulfur bacteria. So we looked at purple sulfur bacteria and purple non-sulfur bacteria. We looked at green sulfur bacteria. Now we're looking at green non-sulfur bacteria. Similar to the green sulfur bacteria, but they use substrates other than sulfides for oxidation because it is non-sulfur bacteria. We have Chloroflexus as an example of a green non-sulfur bacterium. It often has an orange color when it grows in the dark, but it becomes green when it grows in the sunlight. So it stores bacterial chlorophyll in chlorosomes, those vesicle type things in the bacteria, similar to Chlorobium, and performs anoxygenic photosynthesis, so does not produce oxygen, using organic sulfites in low concentrations or molecular hydrogen as electron donors, which is where we get the non-sulfur bacteria part from, using the molecular hydrogen. So it can survive in the dark if oxygen is available. Chloroflexus does not have a flagella, but can glide like Cytophaga. It grows at a wide range of temperatures, 35 to 70 degrees C, thus can be thermophilic. So philic meaning loving and thermo, heat loving. All right. So then we have our large group, another photosynthetic bacteria. So let me back up just a moment here. So here we were talking about our sulfur type bacteria, right? We talk about our phototrophic bacteria is our large category here, phototrophic bacteria. Then we talked about the different sulfur versus non-sulfur bacteria, which are phototrophic bacteria. So the purple sulfur, green sulfur, purple non-sulfur, green non-sulfur. Now we're going to look at a very large category of phototrophic bacteria called the Cyanobacteria. And this is a phylum, the Cyanobacteria phylum. They get their blue and green color from the chlorophyll contained in their cells. Species of this group perform oxygenic photosynthesis. So remember, the other ones mainly were non-oxygenic or anoxygenic. Cyanobacteria, on the other hand, are oxygenic. So they go through oxygenic photosynthesis, which, remember, is where they produce oxygen. And in the case of cyanobacteria, they produce megatons of gaseous oxygen. So they're really great oxygen producers. And in fact, scientists hypothesize that cyanobacteria played a critical role in the change of our planet's anoxic atmosphere 1 to 2 billion years ago to the oxygen-rich environment that we have today. So the scientists really are hypothesizing that these cyanobacteria had a big role because they're able to produce so much oxygen. All right, so here, cyanobacteria, I'll leave the pictures up here for a moment. So cyanobacteria have other remarkable properties. So they are amazingly adaptable. They thrive in many habitats, including marine and freshwater environments, soil, and even rocks. They can live at a wide range of temperatures, even in the extreme temperatures of the Antarctic. They can live as unicellular organisms or in colonies. They can be filamentous, forming sheaths or biofilms. Many of them fix nitrogen, converting the molecular nitrogen into nitrites and nitrates that other bacteria, plants, and animals can use. So they're nitrogen fixers, many of them. The reactions of nitrogen fixation occur in specialized cells called heterocysts. All right, so our cyanobacteria, what they're most well known for is their ability to produce massive amounts of oxygen, but also they are able to live in many different habitats, different types of water environments, soil, even in rocks. They can live at different temperatures, extreme temperatures like extremely cold in the Antarctic. They can live as singular organisms, unicellular, one-cell organisms, or they can live together in colonies. They can be filamentous, or they can form sheaths or biofilms. Many of them fix nitrogen into nitrites and nitrates, which is beneficial for other organisms. So overall, cyanobacteria are extremely helpful and very diverse. So photosynthesis in cyanobacteria is oxygenic, as we've talked about, using the same type of chlorophyll found in plants and algae as the primary photosynthetic pigment. So that's another unique thing about cyanobacteria, is that they have this chlorophyll, the same type that's found in plants, as their primary photosynthetic bacterium or pigment. Cyanobacteria also use phycocyanin and cyanophycin, two secondary photosynthetic pigments that give them their characteristic blue color, so they're kind of green-blue. They're located in special organelles called phycobilasomes and in folds of the cellular membrane called thylakoids. And remember, we talked about that in previous chapters as well, which are remarkably similar to those in plants, the thylakoid in plants. So scientists again hypothesized that plants originated from an endosymbiotic relationship, endosymbiosis of ancestral eukaryotic cells, so some of the first eukaryotic cells, those with a nuclear membrane, and ancestral photosynthetic bacteria. So they hypothesized that something like cyanobacteria, which were photosynthetic, somehow worked together with some sort of endosymbiotic relationship with early eukaryotic cells that then grew to turn into plants. So cyanobacteria are also an interesting object of research in biochemistry, investigating their potential as biosorbents and products of human nutrition. So absorbing things, seeing if they can absorb things for their biochemistry aspect, and then also in human nutrition, seeing if they're beneficial for human nutrition. which even now, a lot of times people will eat powdered cyanobacteria, most likely for the chlorophyll aspect of it. It's kind of like getting chlorophyll from plants. Unfortunately, cyanobacteria can sometimes have a negative impact on human health. So genera such as Microscope can form harmful cyanobacteria blooms. So they form these blooms, which are dense mats on bodies of water, and produce large quantities of toxins that can harm wildlife and humans. So they can grow very fast, grow these really thick blooms, these huge dense mats that float on the water, and then they produce toxins that can then kill wildlife around them and can harm humans. These toxins have been implicated in tumors of the liver and diseases of the nervous system in animals as well as humans. So they can be dangerous, they can be harmful, but most often when we think of cyanobacteria, we think of very helpful. So here is a summarization of our phototrophic bacteria. Here's our cyanobacteria, and then some from the other groups, our proteobacteria groups that we talked about, those that are phototrophic within the Proteobacteria, and then some other ones here as well. The green non-sulfur bacteria, green sulfur bacteria, purple non-sulfur, purple sulfur, and then we have our blue-green bacteria. All right. So the next section that we're going to talk about, so we have talked in two sections now about gram-negative bacteria. We talked about proteobacteria and non-proteobacteria, both of which are gram-negative bacteria. We also talked about phototrophic bacteria, also gram-negative, but includes things from the Proteobacteria group and the non-Proteobacteria group. This next section, we're going to move into the gram-positive bacteria. So we're moving away from all the gram-negatives that we've been talking about, and you can think back to gram-positive with that thick peptidoglycan wall, and we're going to talk about the different groups, the different categories of gram-positive bacteria.